We live in very turbulent, often depressing times. But whenever anyone says to me, what makes you optimistic? One of the top things that makes me optimistic is the fact that we also live in a golden age of science, an absolutely remarkable golden age of science. And it drives me mad that the government and the public in the UK often doesn't recognise what an extraordinary treasure trove of brilliant minds and breakthroughs we have here in Cambridge. So, although AI tends to dominate conversations about scientific innovations right now, and although we are here today to have the 2026 Turing Lecture, named after Turing, who of course is best known for having been the father of AI, we're going to be talking today about something that's actually a twist on that. Because we have the man who actually holds the title of Turing Lecturer at DAMT, who's going to be talking to us, Ray Goldstein, who has had a storied, stellar career. He started off for the first parts of his career in America, at MIT, at Chicago, at Cornell, at Princeton, at Arizona, and then 20 years ago came over to the UK, to Cambridge, where he's been a luminary at DAMT for a number of years. He won the Ig Nobel Prize in 2012 for his work on the ponytail, not to be confused with the Rapunzel number that came out of that work. I'm not joking. So all of you can remember him as the ponytail man. And he asked me, in fact, whether he should go around carrying a chopped off ponytail with him for his lectures to get attention. So that's a teaser. But what he's actually going to be talking about today is the architecture of biological complexity, and in particular, how, as part of this extraordinary golden age of science that we should all be celebrating every single day, biology, physics, and maths are increasingly colliding in some really exciting ways to understand the architecture of organisms. I hope I said that right. If not, your opening remarks can correct what I just said. But in any case, we're absolutely delighted and honoured to have you here today. We're also delighted to have some of Turing's descendants here in the audience, and thank you once again for your support. And over to you to explain to us why ponytails matter and why the architecture of biological complexity needs to be looked at afresh. Thank you.
Thank you very much, Gillian, for that embarrassing introduction. I'll never live down the ponytail, especially when it was a bunch of balding middle-aged men who sat around trying to figure out this problem. Thank you also, all of you, for coming today, and those of you who chose me to give this lecture for the invitation. It's a real thrill to see so many familiar faces in the audience and to see new ones today, and also to have had this very interesting experience of looking through the archive collection of Turing's letters and report cards and other such things. Quite fascinating. So as Gillian said, it is natural in this age of AI to remember and celebrate Alan Turing's great contributions to theoretical computer science, but there's another side of the Alan Turing story which in the wider public is not particularly well known. Perhaps it's known to a number of you in this audience, which is his contribution to mathematical biology, one of really the great contributions in science in the last century or so. And when I was thinking about what to talk about today, I thought I would try to connect the physics, biology, mathematics worlds by talking about a problem that's mostly about geometry. It's mostly about the sort of design, natural design of multicellular organisms, and some of the evolutionary biology questions that go with it. So I won't have too much mathematics in the talk. I realize this is a very interdisciplinary audience, and there'll be a few equations, but not too many. But when I mention the great contribution of Turing to the study of biological pattern formation, I should just start by telling you that this was an amazing paper published in 1952 called The Chemical Basis of Morphogenesis. So by morphogenesis, what is meant is the development of form in biological systems. Now, we're all familiar with either technically or just empirically with the phenomenon of diffusion. You take a drop of ink and you put it in water and it spreads out diffusively over time. And that tells you that diffusion by itself as a phenomenon which shows up everywhere in life at the molecular scale and much larger scales tends to homogenize things. But Turing's great contribution was to realize that the combination of diffusion, which is a homogenizing influence, and chemical kinetics, which tends to be a very nonlinear process involving concentrations interacting with each other, can produce patterns. And this was not obvious at all, but his very simple exposition in this fantastic paper in 1952 showed that this was the case. In the years after this great work, there was a lot of discussion in the physical and biological community as to whether the literal process that he described actually is realized in biological systems. You know, is it what describes your fingerprints or maybe embryonic patterning early on? It was actually not until the early 1990s that physicists actually managed to create an artificial non-biological system where they could test whether this idea was correct. And this group in University of Texas at Austin actually developed a very intriguing chemical system where they could be sure there were no extraneous influences from fluid flow or anything like that. It was a pure system of reaction and diffusion, and it developed spontaneously patterns like you see here, stripes and spots. And in fact, one of the things they showed was that the complex chemical soup that gave rise to this was temperature dependent. And so if you choose as a measure of the order of these systems, something like the difference between the white and black that you see there, that at high temperatures it was basically zero, and as you lower the temperature, it rises continuously, roughly like the square root of the distance from that critical temperature. And this is called a bifurcation in technical terms. And this was the hallmark of what Turing said could happen in a system of reacting and diffusing chemicals. Spontaneous appearance of a pattern out of a homogeneous state. Fantastic result. But if you read Turing's paper, and this is one of the interesting things in the sociology of science, most people read the abstract and a little bit into it, and then they don't read all the way to the end. But if you read the beginning and the end, you realize that he understood very clearly that it was not just about chemical reactions and diffusion, but it was also about mechanics, that the development of form on every scale involves forces pushing and pulling on tissues and rearranging them. And he chose to say, look, I'm not going to talk about the mechanics, I'm just going to talk about the chemical work, but he certainly understood that that was an important ingredient. And about 15 years or so ago, a bunch of physicists started to recognize that actually it really was time to kind of move back toward these more mechanical problems and try to understand the three-dimensional development of complex form, not just the patterns that appear through chemistry. And so there's this very nice homage, Turing's next steps, the mechanochemical basis of morphogenesis. Now, morphogenesis is a very generic term. It can mean many things to many people. And I thought I should just kind of whet your appetite for this by talking about one primary example, which is called gastrulation. How many of you have heard of gastrulation? A few. Okay, so there's a very interesting quote due to Lewis Wolpert. Some of you may know this. He said, it's not birth, marriage, or death, but gastrulation, which is truly the most important time in your life. The thing is that there's no paper by L. Wolpert that says this. Actually, he was apparently talking to a cardiologist in the presence of a friend, and the cardiologist somehow admitted that he didn't know what gastrulation was, and this came out. So what is gastrulation? So gastrulation is a process in early embryonic development in which an initially spherical mass of cells eventually develops into a hollow structure like you see here, and develops an invagination that eventually leads to the formation of the gastric system, and eventually to change the topology of this object from a sphere that it initially was to a torus with a hole all the way through it, like most of us have. Right? And Wolpert's point is that, well, if gastrulation doesn't happen in these early stages of development, well, you're not here. So it is important. Now, it's a very complicated process. It involves cells changing their shape, differentiating into different types of tissue layers, intercalating with each other, migrating around. It's complicated, it's important, and it's the source of great study in the biological world. But you can ask a question, which is, what are the evolutionary precedents of this? Can we look back to simpler forms of life and try to understand how maybe one or two of these processes occur and allow tissues to fold and form the kinds of complex three-dimensional shapes that we know and love in our everyday life? And this question actually leads us to a related question, which has to do with the evolutionary transitions that occurred back billions of years ago that took single-cell organisms and evolved them to become multicellular ones. So if you think of the simplest organisms that appeared out of the primordial soup, single-cell organisms, let's say roughly akin to bacteria, eventually they evolved to become larger in the sense of having more cells, but also more complex in the sense of having more cell types and dividing up life's functions into different cell types. And we have many of those. And there's a natural question, which is, what are the driving forces for that? Why did evolution move in that direction? And that's an important question that links evolution and development together. And I'd like to just show you some data that I think is very motivational for this kind of question. This was put together by Bell and Moores 30 years ago. It's a log-log plot from information in the literature of the number of distinct types of cells found in many different organisms as a function of the number of cells in the organism. Okay, so a bacterium is 1-1. It's one cell, and it has one cell type. It does all the functions of life, which you can think of as reproduction and vegetative metabolic activities. Now, a highly developed organism like a former provost of King's, for instance, is about 1/10 of a cubic meter in size, and a cell is about 10 microns on the side. And if you do the math, that's 10 to the 14th cells, Mike. And you have about 210 distinct cell types. And so you look at this and you think, maybe there's a straight line there, but it's not a very well-defined one. But what you do see is a general trend that as things get larger, they get more complex by this measure. And you see this not only in organisms, but you also see it in human constructs, right? A college has many different types of employees. And because it's complex, it needs all these different types of people to keep running as it does. Whereas, if it's just a school teacher in a one-room classroom, they can do all the functions. But rather than try to encompass all of this at once, it's natural to essentially look in the lower left-hand corner and be a bit reductionist and try to understand some of this complexity by looking at classes of organisms that say, as a function of cell number 1, 10, 100, 1,000, suddenly develop 2 cell types or 3, and try to understand how that happened. And this is actually the theme that underlies much of what goes on in my group in applied math, and actually in the larger scientific community, worrying about the origins of multicellularity. Now, anytime you ask these kinds of epic questions in biology, it's natural to look for what are known as model organisms. So these are organisms that are studied from a variety of different perspectives, ecological, physiological, genetic, et cetera, to get a kind of holistic view of what's going on. And the recent literature, 1700, actually a letter by Mr. Antony van Leeuwenhoek, whose name you might know. He was not the inventor of the microscope, but he was the perfecter of the microscope many years ago. This is an example of his microscope. Reports the existence of a beautiful organism that he found in sheep's livers, gnats, and the animalcula in the excrements of frogs. This is a beautiful organism which is a sphere full of many cells, hundreds or thousands of cells on its surface. And if you read his wonderful prose, it spins around and rotates in a beautiful manner. And it remained unnamed for about 60 years until Linnaeus, in his great book on taxonomy, in the very last entry, gave it the name Volvox, from the Latin volvere, to roll or spin, like the word revolve. It's a fantastic model organism to study these kinds of evolutionary transitions to multicellularity, but it's also a model for the kinds of three-dimensional morphogenetic problems that I want to talk about. So here's an example of what this organism looks like. It's about 800 microns across. So speaking of hair, if you still have any, the typical diameter of hair is about 100 microns. That's 1/10 of a millimeter. So this is almost a millimeter. And it's a beautiful organism with 1,000 cells on its surface. They're called somatic cells on the surface. They have 2 flagella that allow them to swim through their fluid environment. They have daughter colonies on the inside that grow up, and inside of them are the granddaughters-to-be of this mother, which are germ cells that will eventually grow up. And the whole thing is held together by a transparent extracellular matrix, which is a kind of polymeric goo that is elastic and allows this organism to keep this structure. And I'll be returning to this kind of structure in my talk repeatedly. So it's a model because what you see here is that it has 2 cell types. It has what you might call the sterile galley slaves on the outside who just do the swimming and no other functions of life, and the dedicated reproductive cells on the inside, which grow up to become the daughters. So it's got a differentiation between reproduction and everything else. And it's actually related to a single cell organism, which looks just like one of these cells, which has one cell type, obviously. So there's this natural division of labor that occurs. If you watch this under the microscope, this is what it looks like. It's a beautiful, regular architecture. It spins around in a nice way. It was featured a little bit in a documentary the BBC did on the River Tay, a very beautiful documentary. And so here you see it in its natural habitat. This is a real-time video showing you how it spins and moves around. So it's really a nice model for this evolutionary transition to multicellularity. And actually, it's been written about for a long time. It's been understood as a model since the late 1800s. But there's a beautiful book, which I recommend to all of you to read if you're interested, by Julian Huxley in 1912 called The Individual in the Animal Kingdom. And it's a long, fantastic exposition on this question of what are the driving forces to create complex multicellular organisms. And he has this beautiful drawing in the frontispiece, which is a hand drawing, obviously, not a digital photograph like we have now, showing the great structure of Volvox. But this quote is just the one I love. It says, "It was thus the other method, aggregation of similar units and subsequent division of labor among them, that opened to life the full resources of the second grade of individuality. In some colony like Volvox, there once lay hidden the secret of the body and the mind of man." So if that's not justification for working on this, I give up. Okay. So now I'm going to spend a fair amount of time going back and forth between biology and mathematics and physics in this talk. So here's a small section on some biological questions that were raised by Nicole King and Thibaut Brunet some years ago in a very interesting article about the evolution of multicellularity. And they talk about 3 different kinds of organisms. So I've told you about algae. You're all familiar with sponges. You've seen them. You use them. Not kitchen sponges, marine sponges. And if you think about the tree of life, the branch that is animals has as its base sponges. So they are the basalmost animals. And just off to the side of that base are organisms called choanoflagellates. They are the closest living relatives of animals. And in some ways, these cells are similar to cells inside sponges. And what Brunet and King were saying is that there's a very interesting kind of geometrical commonality if you look at these 3 types of organisms. So choanoflagellates in their unicellular form look like a badminton shuttlecock with a flagellum that allows them to swim. So there's a cell that's a few microns across that has a collar of filaments that allows it to filter feed due to the flows created by this undulating flagellum. And as it grows and divides, it can form 3-dimensional structures that often have a very nice geometric regularity to them, but they're held together by something called an extracellular matrix, which is just what I mentioned to you in Volvox. It's this polymeric elastic material. And if you look at the development of sponges, you'll also see a kind of geometry that's similar. The zygote divides multiple times and produces tissues which fold and eventually develop this kind of 3-dimensional architecture, quite similar with a hole in the center here, just like the choanoflagellate has. And if you look at the development of the Volvocales that I was telling you about, eventually you get this spherical structure held together by an ECM. So they were pointing out that there is this commonality and raising the question of what can we learn from that. And after thinking about this for a while, we came to realize there's a kind of unanswered question in biology. I actually hadn't even been asked properly, which is this: how do cells produce structures external to themselves in a robust way? So think of it. These cells are secreting the material that makes up this extracellular matrix. They have no arms and legs and eyes, and yet somehow it self-organizes into a structure. And in some ways, this is very much like what Alan Turing was thinking about when it comes to patterns that self-organize. But it's not just chemical species. It's a 3-dimensional structure. So you can think about something like these green algae on the scale of 500 microns. You can think about these choanoflagellates. And actually, if you think about sponges, this cross-sectional view is what a typical sponge looks like. It's actually a series of spherical chambers lined with filter-feeding cells that are very much like these choanoflagellates, connected together by little tubes. And there's a network, a kind of microfluidic network that stretches from the outside of the sponge all the way to a central chimney, and water flows through it. And all the rest of the space is another extracellular matrix. And my question, which is still unanswered, is how does a sponge start out as a little spherical embryo, which is a sphere, its topological genus is 0, and end up with, I don't know, a genus of 5,400,003 here? What is the process by which you create this complicated microfluidic network? So that's an example of a question. Unfortunately, I don't have an answer yet, but we're working on it. Okay, so I'm going to now segue into a discussion about some aspects of this organism Volvox as a way of motivating this general question of tissue folding and development of three-dimensional form. So the life cycle of Volvox is quite interesting. It's fast, it's 2 days and it's over. So if we pick it up, remember it's a plant basically, it's photosynthetic, it's an algae. So it needs a day-night cycle, and if you grow it in the lab, it's typically a 14-hour day and 10-hour night. And if you pick it up in the morning of the first day, like in Genesis, the daughter colonies will hatch out of the mother, and they have inside of them these germ cells. And during the first day, all that happens is these germ cells grow very large. And then through a very interesting process at night, they go through 9 or 10 rounds of binary division, producing 2 to the 9 or 2 to the 10 cells that will become the next generation. Now, interestingly, they're formed inside out. That is to say, their germ cells, which will become the next generation, should be on the inside, like they were previously, but they're on the outside. And of course, that means that the flagella, which will ultimately be created, are now would point inwards. And I don't know about you, but it doesn't seem logical you could swim with your appendages on the inside. So that problem, that error, is fixed by a fantastic process called inversion, where this sphere turns itself inside out. And I'll show you this in a moment. And after that, all that happens is that the extracellular matrix just expands over time, and then the process continues. So this process of inversion is really quite interesting. And in some ways, in it fixing a problem, it's kind of like gastrulation fixes the problem, that you're supposed to be a torus, but you started out as a sphere. So here are conventional light microscopy views of what this inversion looks like. It's quite fascinating, actually. There's a pure math problem of how to turn a sphere inside out. This is biology's closest approximation to that. So one way or another, some kind of hole develops at the top of this sphere. Usually in this type A case, there's a cross-shaped opening. 4 lips peel back. The whole thing peels around itself and then inverts. And in what's called type B, there's a kind of equatorial invagination that occurs, and a round hole opens up on the top, stretches, and the whole thing inverts. And it does this without any assistance. You know, there ought to be a rule for this. There ought to be some mathematical explanation for how nature manages to do this, and that's what I want to talk about. So if you use conventional methods of imaging such things, you can see beautiful structures at one point in time. So if you use electron microscopy, you can stop this process at any point you wish, and you'll see halfway through there's this kind of mushroom shape in type B, and it proceeds as we saw. Or you can embed this in resin and take slices very thin and look at the cross-sectional shapes of cells. And what you discover is there actually are cell shape changes that drive this whole process. Now, a cell can elongate, and that will take a collection of such cells as a tissue and shrink it this way. It can flatten, and that will expand it. Or there's actually a method by which it can curve a cell sheet. And what we learned from all of these static studies is that what happens in this initial part of type B inversion is that there are connections between the cells that are called cytoplasmic bridges. These are left over from incomplete cell division. Normally we think of cells dividing and just going their separate way. But if they divide and that process is incomplete, you're left with a little tube, which is called a cytoplasmic bridge. And those cytoplasmic bridges are actually connected to filamentary structures inside the cell called microtubules. And along those microtubules are proteins that actually walk and carry cargo. And the cargo they carry is essentially moving the cytoplasmic bridge relative to the cell. And so in this region, all those bridges move to the tip. And so if imagine you had a bunch of bowling pins and you cinched them together at the bottom, they'd flare. And that flaring induces curvature, and that curvature is what you see in the cell sheet. And we know that is the case because there are mutants in which this protein is blocked, and it doesn't happen. So it would be nice to have some kind of quantification of this process to try to develop a mathematical theory. And so what we did in applied math, and I should say, you probably know that rule number 1 in Cambridge is that the names of departments and what goes on inside have nothing to do with each other. And so, yes, it's the Department of Applied Mathematics and Theoretical Physics, but we have a cell biology lab in the basement. And we built there some years ago something called a light sheet microscope. This is not our invention. This is a standard technique in imaging community. The basic idea is you take a laser beam and you put it through a cylindrical lens and you end up with a sheet of light. And you send that sheet of light backwards through a microscope objective, which will make it a very small sheet of light, as you see here. And you suspend your sample in front of it, and then you collect light that comes off orthogonally to it, and you can image a cross-section of the object. And then you move your sample through this sheet, and you build up a three-dimensional stack of images. And here you see a sequence of such cross-sections. Volvox is mostly empty space, but here are the cells on the surface, and just at the end you see the kind of packing of the cells. So when you finally implement this whole technology, you actually can get a three-dimensional video that shows you what's actually happening. And so here are two views of the three-dimensional process of inversion. So you can see over here it happening in various of the daughter colonies, and here it's blown up for you to see, and it's quite amazing, right? There's a kind of ring of curvature that develops here, and just like that mushroom cap you saw in the electron microscope, it moves up, the hole opens up on the top, it stretches around and closes. Right? So it's quite amazing, I think. And once you see this, you really think it's time to try to quantify things. Now, there's no rule book for this. This is not like a highly developed problem in, I don't know, particle physics, where there's kind of a rule book of what you're supposed to do. But we try to quantify it as we would mathematicians. So basically using the same kinds of things. If you had an MRI, you would quantify what you see. You look at these cross sections, you can measure as a function of distance along the curve, the radius, or you can measure how much the bottom has moved up as a measure of how far this process has proceeded, or you can measure the curvature of these regions and ultimately try to connect them to experiment. And so what you see when you try to measure things like the surface area as a function of time is that from run to run, of course, there's a lot of variation. These are small biological systems. There's a lot of noise. But there's a general trend that first there's some contraction, then there's some expansion. That's the top expanding. And then there's a deep contraction as the whole thing finally inverts. And you can measure the curvature in this region, and you see that it peaks up quite nicely as a function of time. So we can quantify almost everything we want. How are we supposed to think about this in some mathematical way? And here I just want to make a few comments about the structure of a mathematical theory. And one thing you can do is make a hypothesis, which is that there's some kind of energy function as a function of shape that governs what we're seeing. And that if I find the minimum energy configuration, that might be the one I see. And that somehow over time I am moving through a quasi-static sequence of equilibria, that the system is just slowly moving in parameter space, and that's what's allowing these shape changes to occur. And the simplest kind of argument you can make is to say that this cell shape change is inducing a preferred curvature locally somewhere on this sheet. So for those who are mathematically oriented, I would say my energy is an integral over the surface of something involving the average curvature, what's called the mean curvature of the surface, minus some preferred value, usually squared. So that would mean if I could everywhere make the curvature of the surface equal to that preferred curvature, I would lower my energy the most I could. There's another contribution that comes from what's called the Gaussian curvature, which has to do with saddle shapes and things like that. We can kind of ignore that. But these kinds of ideas that there's a preferred curvature show up all over in biology. If you look at red blood cells, like you see here in an electron micrograph, they have this biconcave discoid shape. And that can be understood in large part by this kind of energy, where this preferred curvature is set by proteins that differentiate between the outer side and the inner side of the membrane. But if we go to hair, and I were to extract hair from the nearest available ponytail, you would discover that it has built-in curvature. It's not always perfectly straight. There are these natural bends and twists, and a one-dimensional object can pretty much naturally match its local curvature to the preferred curvature, unless that preferred curvature is violently varying. But once you make it a two-dimensional surface, it's not obvious that you can achieve that. Here's one slide on mathematics just to show you briefly how this goes. I won't dwell on it, but the basic idea is that there are two components to a surface. One is that you can stretch things, and the other is that you can bend things. I told you already about the fact that cells can change their shape and expand or contract cell sheets. That's a kind of stretching, and of course they can bend, as I described. So the basic idea is you have some reference state where you think everything is in equilibrium, and then you deform it to some new state, and it has its own geometric representations of where I am along the curve and angles with respect to that. And if I look at the rate of change along my surface relative to this one, I can talk about the stretch in that direction. And if I look at the radius compared to the original radius, I can look at the stretch in this direction. And I basically build a theory that looks like Hooke's law, which says the energy is the square of the stretch relative to its preferred stretch. So my energy turns out to be some integral over the surface of various quadratic forms that are basically in these stretches minus their preferred values. That's the stretching part. And the separate contribution is that there can be bending, represented by the term kappa, minus some preferred value. And that shows up very much like in the theory of red blood cells. So there are two contributions, stretching and bending, and that's what I need to do. So the question then is, if I go through the mathematics of this and ask what are the equilibrium shapes, do they match what I see in experiment? And the simplest picture you would have is that, for instance, when this process starts, the posterior may contract a bit, so the preferred length is smaller there. There may be a region where I get this preferred bend. So if I look at the preferred curvature, there's a sudden drop in that region right near the equator, et cetera. And then I can ask, does this match what I see in experiment? So you turn a large computational crank, and what you see is something what I call the importance of posterior contraction, which is, I think everyone would agree, is important. So for instance, if you imagine that all you did in this problem was have these two little regions in red which had this preferred curvature and nothing else, yes, it would bend inward as you crank up the value of that preferred curvature, but you'd end up with something that's kind of like the purse string effect. You'd be stuck. You couldn't actually get the lower part through the upper part and actually invert the surface, because there's a big barrier to squeeze it through. On the other hand, if you allow the cell sheet to contract at the bottom and you allow, in a temporally adjusted sequence, the top area to expand, then as this happens, you can fit this into that region and actually allow the process to occur. So a spatio-temporally regulated sequence of cell shape changes actually allows the inversion to occur in this quasi-static way. And you can test, if you think that the curvature was actually the other direction, well, it doesn't work either. You get a smiley face and it won't invert. So in the end, you can actually take the observed shapes that you see at the top all during this sequence, and you can fit that kind of energy functional parametrically to what you see and get a quantitative account of the entire process, where you have a wave of cell shape changes that progresses along the anterior, stretching along the exterior. This is again a cross-section. There's even contact going on. And eventually, you can turn the entire sphere completely inside out. Right? So that's, we think, how biology does it. It's a sequence of cell shape changes fundamentally driven by components within the cells that involve stretching and bending. Now, actually, one of the virtues of being a mathematician or a physicist is that you can explore parts of parameter space that are beyond what nature has. You know, that sometimes in the biology community, people will say, yes, but you know, the biological system's like this, and we'll say, yes, but that's part of a much bigger picture, and I want to see the big picture as to what's going on. And the big picture is to think about a parameter space where this quantity k, which is a measure of the depth of that curvature that I'm imposing, is plotted versus some measure of how much the bottom part has moved up. So this is what I observe, d, measured by the radius, and this is what I fix. And I can choose the size of that little region over which I induce the curvature, lambda, to be something, and look at the curve that I get in this parameter space. And for those who have a familiarity with thermodynamics or statistical physics, this looks like a diagram relating the pressure and the volume of a gas, where you actually have smooth behavior when the temperature is high, and below that you get these loops that correspond to phase separation into a low-density system and a high-density system. What happens here that's analogous is the coexistence of two different states with very different parameters, d, in this parameter space, and there's a kind of critical point here. So anytime you're near this kind of instability in the system, you expect there to be lots of fluctuations. And interestingly, what we observe in this inversion process is often a lot of this fluctuation. We call it denting. There are these large-scale fluctuations in the shape just before the shape transformation. And we think that this might be an indication that actually nature is living somewhere close to this instability in the system. So now I'm going to move on to talk about the extracellular matrix. At this point, this organism is just a raft of cells held together by these bridges. There's no extracellular matrix. But the real three-dimensional structure is complicated. It's a compartmentalized extracellular matrix. Around every cell, there's a little kind of domain of protein that forms, inside of which is an elastic material. And around the daughter colonies, there's another domain wall. It's a highly structured, self-organized system. And in fact, if you look with the right kind of microscopy, as in this reference from the web, you can see that there are these compartment boundaries there. And it was actually drawn in that frontispiece of Huxley's book, which shows you it's important to look. But if you look very closely, you can see with the right kind of imaging, there are these beautiful polygonal regions around every cell. And if you do some image analysis using modern AI techniques, basically, you can quantify those polygons and start to ask, how do I reconcile the variation in the size of these domains that you can just see with your eye, the noisiness at the scale of the individual cell, with the great regularity that I see on the scale of the whole organism. And that's a central theme in much of biophysics. So this brings up an interesting point, and also a point about taking up opportunities in science when they come your way. Right in the middle of the pandemic, we were contacted by the group of Will Ratcliffe at Georgia Tech, who had been studying something that Ratcliffe pioneered in his earlier PhD work in Minneapolis. And it involves the discovery, or the creation, let's say, of an organism now known as snowflake yeast. So yeast, to most working biologists, is a unicellular organism. You grow it up in solution, it happily lives as a single cell, feeding off its environment. And this amazingly important experiment by Travisano and Ratcliffe et al. Asked the question, could we force a system to adopt a multicellular lifestyle by some selective pressure? And so they did the following experiment. You grow yeast up in a test tube until it's a certain concentration. You put it in a centrifuge for a period of time, and you centrifuge everything you can to the bottom, and you throw off the top 95%. You take the 5% at the bottom, dilute that into a new growth medium, grow it up, and do this over and over and over, always centrifuging the system and selecting for the bottom. And what you discover after 20 or 30 or 40 or 50 rounds of this is that what's at the bottom is no longer unicellular. It's actually these kinds of ramified structures that you see here that look kind of like a snowflake. Now you might think, well, these are just unicellular cells that are sticking together. But it's not the case, because when this organism divides, it produces multicellular progeny, which is just amazing. And now, after many years of studying this, they've been able to show that there is genetic differentiation between the periphery and the center. And so it's really starting to develop into a multicellular organism. So by this selective pressure, they've turned on this lifestyle. It was kind of always there, but in such a small amount that you could never see it. So this group contacted us because they were working with physicists at Georgia Tech, particularly Peter Juncker, asking the question about the geometry of these organisms. And the way they studied it was essentially what is called a Voronoi tessellation. So imagine you have a bunch of cells, in this case these yeast cells, and you take the center of each cell and you look to your neighbor and you draw a line there and you construct the perpendicular bisector of that line as a plane, in this case, and then you do that with all your neighbors and you end up with a polygon around every cell. And then you ask, what is the distribution of the volumes of those polygons? How random is the system, or how regular is it? And they found a certain law and were interested in whether we had any data on a different kind of organism, whether it might conform to the same law. Now, snowflake yeast basically appeared yesterday on Earth, and Volvox appeared, I don't know, 500 million years ago. They have utterly different reproductive dynamics. They're completely unrelated. But the amazing thing we found together with them is that they share a common law for the distribution of these cellular neighborhoods. And so if you measure, in the case of snowflake yeast, the probability of finding the volume of those Voronoi cells normalized by the volume of an individual cell, or in the case of Volvox, you measure the area of one of those polygons on the surface relative to the cellular area, you'll discover that the distribution is a very broad, skewed distribution. So it's not just a very sharp, narrow kind of thing that you'd have if you had a perfect hexagonal lattice of these things packing together. It's a very broad, skewed distribution. And mathematically, it has a form that looks like this: x to the k minus 1 e to the minus kx, where x is some normalized volume. Now, this is known as a gamma distribution. It shows up all over the place in science. And it was just not obvious why this should appear here at all. But the fact that it shows up for these two different systems suggests there's something deep going on here. And I'm going to give you a few hints about what might be occurring and why we'd see this. But you see that it shows there's a tremendous amount of noise, a tremendous amount of variability at the individual cell level. And yet there's regularity on the large scale, particularly for Volvox. So you hunt around for a while, and then you discover something important, which is, there are three important methods in mathematical physics that I learned. One of them is adding 0. One of them is multiplying by 1. And the third is mapping it onto a previously solved problem. That will get you very far in science. And so actually it turns out that just next door, in pure math, there's a whole community of people who were worrying about problems that effectively boil down to this. Take a surface, throw down a bunch of points randomly by some rule, which is uniform over the surface, so you're not biasing it, and then construct a Voronoi tessellation of those points and ask what is the probability distribution of areas. This is very extensively studied, particularly numerically in 2 dimensions. And what you generically find is it's exactly this gamma distribution. So that's kind of weird, but it starts to tell you something about what might be actually driving all of this. Because the typical rule that you apply here is called a Poisson distribution, where the probability is related to an exponential in area. So these results suggest that there's something rather generic about these problems where you overlay noise on a system and distribute them in space. But the thing to remember here is that in the case of this growing Volvox, the cells are the source of the material and they're the template at the same time, which really makes it hard to think about mathematically how to formulate it. But apparently a snapshot looks just like these problems. So here's another small piece of mathematics that just shows you what could be going on. There's this common joke in physics about a farmer who's having trouble with his cows, and he asks the physicist for help, and the physicist says, well, consider a spherical cow to begin with, because it's simpler. Right, so let's consider a linear Volvox. Right? Never mind that it's a sphere. Just imagine it's a bunch of cells that are spewing out material according to some rule, and I'm going to construct the Voronoi tessellation of that problem. So imagine that X and Y represent the amount of material in successive cells, indicated by these hashed lines here. And each of them is distributed probabilistically exponentially. So there's some probability of sending out X worth of material that's exponential in X. So the rules in probability tell you that if you first add the 2 amounts of material together, the probability distribution of the sum is the convolution of the 2. And from this rule, you'd actually get this function. And if you divide by 2, which you have to do for the Voronoi tessellation, then there's another rule. And when all is said and done, the probability distribution of the Voronoi cells of this is a gamma function with k equals 2. And in Volvox, we measure 2.3. So already it starts to tell you that perhaps what's going on here is that we're looking at the export of proteins from these cells in a random fashion, perhaps governed by some kind of exponential, and that's what's at the origin of this kind of distribution. So in fact, if you think about emitting B bursts of proteins, you can actually recognize that there's a relationship between the K you measure and the B you measure. In the case of bacteria, this bursty production of proteins is well known. It's been studied in a beautiful set of experiments dating back almost 20 years. In which one measures by fluorescence techniques the amount of protein being given off by bacteria by virtue of it sticking to the cell membrane. And the typical number of bursts that you see per cell cycle is small, and the probability of the number of molecules per burst is exactly exponential, just as you expect. So what this suggests is that if you kind of go back to the central dogma of biology, what we have here is a gene producing mRNA leading to proteins being exported from the cell, and as long as the degradation rate outside the cell is much lower than the degradation rate of the mRNA, it corresponds to bursty production. That might be what we're seeing. Okay, now remember that a Voronoi tessellation is a human construct. I just take a bunch of centers and I just lay down this thing. So there's several directions one would like to go. First, one would like to have a better idea of actually what's going on with this internal structure in the extracellular matrix. And at the same time, you'd also like to finally have some real growth dynamics associated with these problems. So in very recent work by my postdoc Jane Chui and Anand Srinivasan, who's a PhD student, we've succeeded in being able to sort of multiplex our studies of these growing cells by having 6 micropipettes that actually hold an individual Volvox in a confocal microscope. And we, over the course of days, we cycle between one and the next and get successive dynamics of this growing structure. And you can now see, in this case, over the course of about 2 days, an individual colony grow. So here are the daughter colonies. Well, actually, here are the germ cells that haven't yet started dividing. This is one frame every 12 minutes or so. So watch the cell divisions here, and then watch the inversion. Boom. They all go invert. And the sphere just keeps growing and growing. And so out of all of that noisy extracellular matrix production, there's this beautiful regularity. And that's really what we're trying to get at. I don't fully have the answer, but at least we have some data to think about. The second thing is that thanks to an incredibly talented group in Germany under Armin Hallmann in Bielefeld, there's now a phonetically engineered strain of Volvox in which one of the key proteins in the extracellular matrix is fused with a fluorescent tag, so you can actually see it. It lights up. And so this is what you can see. This is an image of essentially the whole organism. Magenta are the somatic cells, which have chlorophyll, and you can induce them to fluoresce naturally. They're shown in magenta. And then all the compartments around each cell show up in green. And you can take cross-sections and see all the three-dimensional structure. It's really like, it's sort of cheating. Now you see everything. And you'd like to be able to understand what's going on there. And one of the things we discovered, actually, is that there's an incredible coincidence between the actual compartment boundaries and what we would construct by a Voronoi construction. So the Voronoi just says, find the midpoint, develop a polygon around you by that. But when you look at this foam-like arrangement and you overlay the Voronoi tessellation, it's incredibly close. So what we think that's saying is actually that this boundary that we're seeing is actually essentially emanating from each cell, and they're colliding. When they meet, they collide and form this polygonal structure. But if you wanted to develop a Voronoi tessellation, you would launch two things and have them collide in the middle, and that's how you do it. And actually, you know that there are two because at another stage of development, if you look carefully, you actually see that whereas this structure just looks like a single wall, it's actually a double wall because each cell has a circular domain which is ultimately going to collide. And so this tells us perhaps that the way to move forward here is to think about the compartment boundaries as kind of what we would call gelation fronts, a polymer network that's essentially precipitating out as a rigid object colliding in the center. And this would provide an explanation for this near Voronoi behavior. Okay, one last thing to say that's kind of amusing is that if you mechanically break up these colonies, just with hydrodynamic shear, you can break them up into pieces. And one of the things you discover is they spontaneously curl up into things that look like seed pods. And you can see here, they still swim. So this is a high-speed video slowed down for you, but the whole thing is rotating because of all those flagella that are pushing on the fluid. And again, you might think that something related to Hellfrisch's elastic energy could play a role here, where this curling is a sign that there's an intrinsic curvature in the system that's now freed to make the organisms curl up. And so based on all these measurements of the growth, we can actually see very carefully that over time, for many different runs and suitably averaged, there's a perfectly linear growth of the average radius of these organisms. There's slight diurnal variations around them that are natural for anything doing photosynthesis. But this linear variation is very consistent with just water being sucked in by a matrix, this extracellular matrix, and swelling. Okay, so now I want to switch gears to the final subject of my talk, which is going back to these choanoflagellates. And here I want to talk about an amazing discovery that was made by Nicole King's group back in 2019 of a form of choanoflagellates that had not been seen before. So let me remind you that choanoflagellates are these wonderful shuttlecock-shaped organisms. And this particular one, called Choanica flexa, appears as a raft of cells. It was discovered on an island in the Caribbean on a field trip, as one does, if you're a biologist, and in tidal pools, basically, on the beach. And this amazing organism, as you see here, can exist in two different forms. So here are the individual flagella beating along happily. It's a raft of cells held together by adhesive contacts between the tips of those microvilli. So think of it just as a glued together collection of cells. But what you're seeing here is actually a bowl-shaped organism in which, in this particular configuration, the flagella are all pointing in. And the raft is rather sparse, let's say. But if you change the lighting conditions, or give it caffeine, then actually it can induce a transformation to the opposite curvature. So whereas the first one had the flagella pointing in, this one flips around, and now the flagella are pointing out, and the cells are tightly packed together. And it just goes back and forth between these two. And amazingly, this one is very poor at swimming, but very good at filter feeding because the cells are wide apart. And this one is very poor at filter feeding, but very good at swimming. So when you change the light conditions, it responds by being able to suddenly swim and go somewhere better. So when we learned of this, we immediately asked Thibaut Brunet and Ben Larsen to come visit because we thought, this is so similar to what we're seeing with this Volvox inversion. Maybe we can work on some sort of mathematical description of this and ultimately understand a bit about the mechanics. So here again are these two pictures. If you look carefully in the right plane of microscopy, you can see the individual microvilli. And if you color code the polygons that you see from this, where the tips are adhering, you see that it really is just a collection of polygons, maybe some heptagons, hexagons, pentagons, etc. And what's going on actually is that the color has two different states. And when the biochemistry of the light changes, the color changes its opening angle. And this is what's biasing the system to flip from one state to another. So how do you make a mathematical model of this? You know, it looks incredibly complicated. We tried to summarize it in the following way. We said, let's take a bunch of spheres and some representing cells and connect them together by the minimal set of connections you'd imagine. So each microvilli, each collection of microvilli will just be replaced by a single one going to each neighboring cell, with some spring allowing it to deviate from some preferred angle, and some linear spring here allowing it to stretch a little bit. And we'll distribute the points on the sphere by taking what's called a geodesic icosahedron, which is just a regular polyhedron, and projecting the points onto the sphere. And you can do that in such a way that they're pretty regularly spaced. So you do this, and then using perfectly linear elasticity, Hooke's law, everything is just force proportional to displacement, you can show that if you change the opening angle as the color does, that you can invert one bowl-shaped structure into another, just like they see in experiment. And we thought, oh, good, we're sort of done. The problem is, where are these two different curvatures coming from? All I did was use linear elasticity, and in linear elasticity, there's no way to get these two competing stable minima. In fact, if you construct the energy of this system, you discover it has two different branches as if they're two competing minima, and that's not part of linear elasticity. And I really struggled to understand, how did we introduce the bistability, the fact that there are two competing structures, in this system? It must have happened automatically. How did it actually occur? And then I remembered something about what are called topological defects. So let me give you an example. So if you look at another example of green algae called gonium, here it is 16 cells, you can ask yourself how this extracellular matrix connects every cell to every other cell. And if you look carefully, which I only did after years of seeing this image, you actually discover that whereas you might have thought that things should be connected to 6 of their neighbors in the plane, this particular one has 7 neighbors. And if you play around for a while, you can realize that having either 5 or 6 or 7 neighbors instead of 6 tends to introduce a preferred shape to the system. And gonium, for instance, as you can see in this beautiful video, is actually a saddle shape. It's not flat. And there's an obvious hypothesis that there's a connection between these 7-fold and the saddle, which reminded me of a very important paper by David Nelson and Sebastian Sung many years ago, who were asking themselves about the structure of networks that were basically hexagonal with a defect of one sort or another introduced. So imagine that you replace one of the cells in this thing by a pentagon or a heptagon. And they weren't the ones to invent this, but they studied mathematical elastic properties of this system. If it's a pentagon, it tends to produce curvature, like a bowl. And if it's a heptagon, it tends to produce a saddle. And you know about this because if you play football on this side of the Atlantic, right, the soccer ball has pentagons and hexagons. And the only way you get the curvature is you have the pentagons. So I looked at their mathematical analysis, and I kept thinking, I don't understand where this bistability comes from. I do have a prop. I don't understand where this bistability comes from because I look at their analysis and it's like not obviously there. But somewhere in their analysis they said, well, imagine a flat sheet, and now I'm going to allow it to perturb upward, and I'm going to calculate the energy. So the energy is a combination of stretching and bending. And then I realized, well, nobody said you couldn't just imagine bending it down. And in fact, if you construct a molecular model, as I did here, the kind you use in chemistry classes, right? You can introduce some pentagons, and you'll see it naturally curves. But if I had disassembled this thing partially, I could have constructed a completely energetically equivalent shape in the other direction that would just look like the mirror image. So there's nothing to distinguish up from down as long as you've got pentagons. So, okay, so the bistability is an intrinsic feature of having pentagons. And if you're mathematically inclined, that means basically the pentagons introduce Gaussian curvature, which is the radius, one of the radius squares, so it doesn't care about the sign. So we thought, okay, this suggests that there ought to be a lot of pentagons in C flexa. So we asked Thibaut and Ben, can you measure the distribution of pentagons in your organisms? So they went through their confocal slices, and they found the right plane in which to look at this, and discovered that something like 22% of the polygons that they saw are pentagons. So the fundamental equivalence, near equivalence of these two states is a consequence of the fact that nature built this thing with lots of pentagons in it. And that's the lesson to learn. But Thibaut's now at Institut Pasteur in Paris, Thibaut's group has done something even better. Very recently, they published a paper where they showed that if you gently agitate this colony, you can break it apart, but not in any way that does permanent damage. It will reassemble itself. And when it reassembles itself, it's perfectly functional. It must have all those pentagons. It can invert, it can do all the phototaxis, all that kind of stuff. And we've been very interested in this. And so with my postdoc, Satyan Anand, we've started to be able to study this process quantitatively. So here's a video over time through the microscope where we're watching, this is one frame every minute for 6 hours. And what you're seeing is the slow emergence of all of these rafts of cells. Sometimes they merge with each other, but the individual cells manage to find this raft, accrete to it, grow this raft of cells in just such a way that there's the right distribution of these topological defects for the system to have this ability to flip back and forth. And I don't know about you, that's a mystery. How on earth do the cells know what to do to make this happen? And so I think this is a good place to stop as an example of the mysteries that still exist when it comes to understanding morphological transitions, development of form in biology. And with modern techniques of imaging and the meeting of physics, math, and biology, perhaps we'll finally be able to answer a question like that. So thank you very much for your attention. Happy to take questions.
Mike already? Mike has a question. Mike has the mic.
Well, I have two, actually. Can I ask them both? The first is that you talked about the evolution of these structures as minimizing some free energy, but you showed pictures of vibration during this process. It doesn't seem to be consistent.
So think of it as a noisy energy minimization problem. So there are fluctuations around the local minimum. In this case, it's not thermal at all. It's driven by the active processes in the cell. So it's not that it's a mistake. It's that actually, at least for these systems, it appears that there are often small fluctuations on top of these local minima. But that's not always true. In fact, if you look carefully at the inversion process itself in the ball box, there's tremendous amount of fluctuations. And it's not at all obvious that you should think of it as quasi-static equilibria. So that's a hypothesis. In some sense, I'd love to prove it wrong.
Yeah. So the second question is, you showed these pictures of these things turning inside out. At that moment, are all the cells the same? What's the difference between those at the top and those at the bottom, the ones that expand and the ones that contract?
So what's happening
They already evolved as different.
Well, they're different in the sense that they're transiently different. That is to say, the ones in the posterior have transiently changed their shape. And actually, there's a wave of such cell shape changes that's occurred.
And who did that to them?
Yeah, and just to complete it, and the ones at the top have become squashed this way, and so they've expanded the sheet. So in many biological systems where you have this kind of symmetry breaking, there's some, well, to use Turing's language, there's some morphogen, there's some chemical species which has a gradient or spatial variation which is telling this one do this one do that. We have, as far as anyone knows, there's no such morphogen in these systems. No one's ever found it. But that doesn't mean there isn't something equivalent to that, in the sense that when you go through these multiple rounds of cell division to produce this bowl-shaped daughter colony, there's an age difference between them. So it could be that basically there's a kind of mapping between age and behavior that is going on. Or it could be mechanical stresses, which are giving cues that are different in the different parts.
So when you have the hexagons and the pentagons in your bowl, do you think that it's forming pentagons because of some force that's making it curved, and then the pentagon is the way to minimize the energy? Or do you think it's forming the pentagon and then that's causing the curvature to be the best way to minimize the energy? Like, do you think the pentagons are cause or the effect?
Chicken, egg, yes. So my best guess is that the particular geometry of the microvilli contacts is introducing a general tendency toward curvature. And this manifests itself as occasionally enough stress building up that the natural way for the next one to fit in involves a pentagonal neighborhood. I think that's the case. And the evidence Thibault's group has and our group has is that cells come down from above, let's say, toward the raft, and they wander around, essentially, I think, anthropomorphically, trying to find a place to fit. And so there's some kind of exploration of possible neighborhoods, and they are latching into the most favorable one each time. And so if you're going to continue to curve like that, you're naturally going to open up spaces that are appropriate for pentagons, I think.
Thank you so much. So you start with gastrulation, and then we saw things similar to that. But I'm curious, after seeing Wallbox and all these things that sort of flip inside out, do we know why it doesn't happen in the gastrulation phase, and that gap basically remains open, and that, you know, leads to a lot of wonderful things happening afterwards?
I'm not sure I fully understand the question. So in gastrulation, you develop a topological transformation in the sense that eventually you develop a hole through and change into a torus. And what was the distinction you were making?
So in my like untrained eye, looking at gastrulation, it looks similar to when a wallbox, before it flips inside out, it forms this similar kind of inverse gap, right? But then the gap doesn't remain that way because it continues going up, and then there's this tearing happening in the outside, and that doesn't happen in gastrulation. The hole remains there, and then
So I think I would say two things. One is this famous article, which was entitled "Nothing in Biology Makes Sense Except in the Light of Evolution." So, I mean, you have to think. I mean, there's the mechanistic, mechanical differences between the systems, but also the evolutionary pressure, which has driven the system to be selected to go a certain way. So I actually don't, off the top of my head, I wouldn't have a clear answer to which mechanical feature allowed one to progress one way or another. But I would say that, you know, there's an evolutionary pressure for millions or hundreds of millions of years which has said stop there or don't stop there. But remember that gastrulation is full of a lot more complicated processes. It's got multiple tissue layers. It's got all these other processes occurring which can regulate things in ways that are not possible here. Also, the tissue is confluent in the sense that the cells are just touching each other. There's no extracellular matrix. Here, it's really these independent players connected by the bungee cords, but not in the same way continuous as in gastrulation.
Can I ask what you mean by energy in this process? So you're talking about the energy of which bit? As a core biologist, I'm struggling to understand energy here. I think you mean the energy in the structure, right? Because, of course, as a biology, we use energy in a different way, and we talk about minimalizing. We see energy as a cost. And so one of the things I'm looking at this, you've got a packing problem, and you get imperfections in this packing problem. It would cost a lot of energy for a cell to ensure it packed exactly the same every time. And then you get these lucky circumstances which evolution picks on. And so I'm wondering which energy is minimized.
Well, this is not a statement that this is the most efficient of all processes that could occur. This is a statement that the shape that I see, given for instance, given the shapes of all the individual cells. If I told you the shapes of all the individual cells and I told you how they were connected, so I could infer from that what they'd like to do as far as bending the shape. If I ask, then if I let it go and find the optimum such shape, is that what I see? But that's hiding a lot of non-equilibrium processes, because the process of elongating the cell with microtubules, et cetera, uses ATP because it's driven by motor proteins. So there's a lot of energy input into the system, dissipation essentially, to create these structures. And I'm looking at a larger scale, and I see it as a system endowed with an energy that I can minimize and find the shape. But for sure, it's consuming a lot of free energy as it does this. Yeah. So it's it's a language problem on some level, but there's a real physical difference there. That I accept that there are non-equilibrium processes going on, but there's an equilibrium that underlies it. You see this a lot in pattern formation. I mean, there are many examples of Turing instabilities in the chemical world where you cannot assign an energy to the problem. There's no sense in which what you see is the minimization of an energy. But there's some problems for which it is. And so even though it's driven by chemical kinetics and diffusion and all that, there's actually an identifiable energy which is being minimized. It's not always the case, but sometimes. So it can be either.
Did you have a question?
Hello. So thank you for that wonderful lecture. It's very enjoyable stuff. So a few slides ago, I think you had it was an algae where there was a cell with 7 neighbours, and you commented on it being a sort of saddle shape. And then you immediately moved away and started talking about hexagons with the occasional pentagon, like the model you sat next to, and immediately made this sort of sensible comment of it being like football. So, in other words, spherical shape. Now, the mathematician in me thinks, hang on, he's saying saddle, that's hyperbolic geometry, and the curved things like that are more like spherical geometry. So, are there biological reasons why, say, one of the algae preferred a hyperbolic way of doing things, and other things preferred a more spherical way?
Funny you should ask that. So actually, literally over the last week, we've been watching this organism, gonium, grow and trying to quantify what it does. And I'm starting to think actually that this heptagonal argument may not be true in the sense that what you discover is that among those 16 cells, there is a differential growth between the ones in the center and the ones on the outside. And you end up actually with, well, mathematically we call it a non-Euclidean metric. There's just too much material to keep it flat, and something has to happen. And so it buckles, and it naturally buckles into a saddle shape. And then you might ask, well, why? What purpose does it serve? And actually, when you watch the reproduction of the organism, each of those 16 cells has to produce a 16-cell daughter. And that's a hard problem to fit everything in. But if you make a saddle, there's a lot of extra sort of surface area in there. And it looks to us, when you finally see the whole thing three-dimensionally, that actually this is a good way to make room for all that extra material. So I think it could be that kind of argument that's actually what selects, at least in some cases, that hyperbolic geometry.
Thank you very much, Professor Goldstein. Really interesting lecture. I noticed somewhere in the middle you show 3 species in the various morphological changes. The number of segments in each one was a prime number. Is that anything to do with the energy function that they're minimizing? 7, 11, and 23.
I have to confess, I didn't notice that.
I need to get out more.
But these were the drawings you meant. Yes. So that may be a human accident, but I don't think that prime numbers are playing a role here. I could be wrong. I don't think they're playing a role because actually there's a lot of variability in the numbers from realization of an organism to the next. But prime numbers, of course, show up in other kinds of problems in biology, but not in this, I don't think.
Okay.
It's a good idea, though. I will check.
Thank you so much for such an extraordinary talk. It was absolutely captivating, and I love all the graphics you've been. It's so good. And I'd like to say a few other thanks. First of all, to the Stanley Foundation, who support the Turing lecture, and we are very grateful for their support, and also for all of you for coming along, for being engaged, for asking questions, for supporting the community here at King's, which is just such a wonderful place to be. And thank you very much, Ray Goldstein, Professor Goldstein. It was a lovely talk.
Thank you.
So, yeah.